The overall goal of this procedure is to produce recombinant parvoviruses, or RECPVs, at high titers while preserving their quality and reducing production times and costs. This is accomplished by first producing an initial RECPV viral stock through transfection of HEC293 T cells with RECPV plasmids together with an adenovirus type 5 derived vector. Since the introduction of add genomic elements into the HEC293 T cells increases the production of RECPVs by more than tenfold in comparison to existing protocols, a novel AdVP helper was generated that contains the genomic adenoviral elements necessary to enhance recombinant parvovirus production, as well as the parvovirus VP gene unit. The next step is to produce and purify this novel AdVP helper according to standard protocols. The final step in this procedure is to amplify the RECPVs by co-infecting NB324K cells with the RECPV and AdVP viruses. The efficiency of RECPV production is improved by more than 100-fold with this strategy, which can be used exclusively once a small amount of RECPVs has been produced via transfection. Ultimately, quantitative real-time PCR, or a transduction assay,